Unit 7 Bioinformatics. In this unit, we are going to study bioinformatics, which is a very important and scoring topic for GAT-B 2026. Bioinformatics mainly deals with using computers and software tools to understand biological data like DNA, RNA, and proteins. In today's time, huge amount of biological data is generated, so without computational tools, it is very difficult to analyze and interpret this data properly. For GAT-B exam, this unit is very important because questions are mostly concept-based and direct. If your understanding is basic terms like databases, BLAST, alignment, and phylogeny, you can easily score marks. So in this lecture, we will cover all important topics step by step in a simple way for better understanding. This slide is mainly about the learning approach and how preparation should be done. The focus is on concept-based learning instead of just mucking up facts. For GAT-B, this is very important because questions are not always direct. They test your understanding of concepts. Also, practice of MCQs and previous year questions is very important. Regular revision, mock test, and doubt solving will help your performance. So, overall, the strategy should be simple: strong concepts, regular practice, and proper revision. Major bioinformatics resources and search tools. In bioinformatics, we use different types of databases to store and retrieve biological data. Primary databases like GenBank, EMBL, and DDBJ show raw sequence data, while secondary databases like PFAM and ProCyte store processed information such as protein families and domains. Composite databases like RefSeq provide curated and non-redundant data which is more reliable. For searching sequences, tools like BLAST and FASTA are very important. BLAST is most commonly used because it is fast and gives local alignment. FASTA is also used but slightly different. Advanced tools like PSI-BLAST and HMMER are used for deeper analysis. In GAT-B, you must remember difference between these tools and their applications. Sequence database show nucleotide and protein sequences along with their annotations. Primary databases like GenBank contains raw and redundant data, while curated databases like RefSeq provide high quality and non-redundant sequences. This difference is very commonly asked in exams. Protein databases UniProt is also very important, which has two parts: SwissProt reviewed and TREMBL unreviewed. Other databases like ENSEMBL are used for genome information, and GEO is used for gene expression data. Always remember, accession number is used to uniquely identify sequences. Structured databases store 3D structures of biomolecules like proteins and nucleic acids. The most important database is Protein Data Bank (PDB). As these structures are obtained using experimental techniques like X-ray crystallography, NMR, and cryo-EM. These structures help us understand protein function, active sites, and also help in drug design. Tools like PyMol and Chimera are used for visualization. In GAT-B, questions can be asked about structure determination method, and uses of structural databases. Biomolecular sequence file formats. Different file formats are used to store biological sequence data. FASTA format is the most common and simple, where we have a header line and sequence. GenBank format is more detailed and includes annotations and metadata. Other formats include GFF or GTF for genome features, SAM or BAM for alignment data, and VCF for mutations and SNPs. Each format has a specific use, so you should not confuse them. In GATB, questions may come from on identifying these formats and their applications. Scoring matrices are used to evaluate how good an alignment is. The two types are PAM and BLOSUM. PAM is based on evolutionary changes, while BLOSUM is based on conserved region of proteins. In these matrices, positive scores indicate good matches and negative scores indicate poor matches. Gap penalties are also added for insertions and deletions. For example, remember that PAM is used for closely related sequences and BLOSUM for distantly related sequences. Sequence alignment is the process of arranging sequences to find similarity. There are two main types, global alignment and local alignment. Global alignment aligns entire sequence, while local alignment finds best matching region as given. Tools like BLAST and FASTA are used for fast alignment, while Cluster omega and muscle are used for multiple sequence alignment. Alignment is very important for finding homology and predicting functions. In GAT-B, you should clearly understand difference between global and local alignment. Phylogeny is the study of evolutionary relationship between organisms. It is represented using phylogenetic trees, which can be rooted or unrooted. Methods like UPGMA and neighbor joining are distance-based, while maximum parsimony and maximum livelihood are more advanced methods. These trees help us understand how organisms are related and how they evolved. Bootstrapping is used to check reliability of trees. In GAT-B, questions are often focused on methods of trees construction and interpretation. Data mining in genomics means extracting useful information from large genomic datasets. It is used for gene prediction, SNP detection, and genome annotation. Machine learning techniques are also widely used in this field. Databases like EMBL and UCSC Genome Browser are used for genomic analysis. Techniques like clustering and classification help in identifying patterns. This topic is important for understanding modern biology and is relevant for GAT-B exam from application point of view. Proteomics is the study of proteins, including their structure, function, and interactions. Tools like mass spectrometry are used to identify proteins. Databases like STRING help in studying protein-protein interactions, while tools like InterPro and PFAM are used for functional annotation. Protein structure predicting tools like AlphaFold are now very important in research. Techniques like ITREQ and SILAC are used for quantitative proteomics. For GAT-B, you should remember major tools and their applications. as questions are usually direct from this topic. Model dynamics is used to study movement on biomolecules over time. It is based on Newton's laws of motion. In this method, we stimulate how atoms in proteins or DNA move and interact with each other. Forces fields like amber and are used to define interactions between atoms. The stimulation includes steps like energy minimalization and equilibration. Applications include protein folding and drug binding studies. In GAT-B, we focus on basic concepts and terminology. Force fields are mathematical model used to calculate energy of a system. They include bonded interactions like bonds and angles, and non-bound interactions like van der Waals and electrostatic forces. Common force include like OPLS and GROMOS. These forces field are important because they determine accuracy of stimulation results. However, they have limitations and cannot fully represent quantum effects for GAT-B. Remember, components of force fields and examples. Protein-protein interactions are interactions between two or more proteins, which are essential for many cellular processes like signaling and metabolism. According to the slide, these interactions can be transient or permanent. They involve forces like hydrogen bonding, ionic interactions, and hydrophobic interactions. Experimental methods like yeast-2 hybrid are used to study them. Databases like String store interactions data. In GAT-B focuses on types and methods of detection. Protein-nucleic acid interactions involve binding of proteins with DNA or RNA. These important processes like transcription, replication, and repair. Common DNA binding motifs include helix-turn helix, zinc finger, and leucine zipper. These interactions are mainly due to electrostatic and hydrogen bonding forces. Techniques like CHIP, SEQ are used to study them. Mutations in these interactions can lead to diseases. For GAT-B, remember motifs and their functions. Protein-ligand interactions involve binding of small molecules to proteins, which is very important in drug discovery. According to the slide, these interactions can be reversible or irreversible. They involve forces like hydrogen bonds and hydrophobic interactions. Binding affinity is measured using values like Kd and Ki. Tools like Autodoc are used for studying these interactions. In GAT-B focuses on types of interactions and applications. Molecular docking is a method used to predict how a ligand binds to a protein. It involves two main components, search algorithm and scoring function as mentioned. Docking help in drug discovery by predicting binding oriented and energy. Tools like AutoDock and Glide are commonly used. However, accuracy depends on scoring functions. For GAT-B, remember basics of docking and its applications. Molecular dynamics stimulation follows a step-by-step workflow. First, we prepare the structure, then select force field and add solvent. After that, energy minimalization is done to remove classes. followed by equilibration step like NVT and NPT. Finally, production run is carried out and results are analyzed using parameters like RMSD and RNSF. This workflow is important for understanding how stimulations are performed. In GAT-B, questions may come in sequence order of steps. Genomic data analysis involves tools used for analyzing genome sequences. Genome browsers like UCSC and EMBL are used for visualization. Alignment tools like BWA and Bowtie are used for NGS data. Gene prediction tools like AGUSTS are also important for GAT-B. Remember function of major tools. Proteomic data analysis focuses on analyzing protein data using computational tools. Software like MaxQuant and Proteome Discover are used for processing mass spectrometry data. Protein identification is done using peptide mass fingerprinting. Databases like UniProt are used for reference. PTMs like phosphorylation are also studied. In GAT-B, focus on workflow and important tools used in proteomics. Systems biology is an advanced approach where we study biological systems as a whole instead of individual parts. It integrates data from genomics, proteomics, and metabolics to understand how different components interact with each other inside a cell. Tools like Cytoscape are used to visualize biological networks, and databases like KIGG help in pathway analysis. This approach is very useful in disease research and drug discovery. For GAT-B, focus on concept of integration and network analysis terms like hubs and pathways. This slide is very important for revision before exam. As shown in the slide, you should clearly remember differences like primary versus secondary databases, BLAST versus FASTA, and PAM versus BLOSUM. There are very commonly asked questions. Also focus on alignment types, phylogenetic methods, file formats, and basics of molecular dynamics. Interaction types and docking versus stimulation are also important. If you revise these points properly, you can easily score good marks in bioinformatics section. Subscribe Brainzz for consistent study, strong concepts, and regular revision.